to introduce Professor Shan Ehlert, who is going to talk about the HI genetics. And I would like to emphasize how important the work that Professor Ehlert is, not only for the children of England, but the children of the world, because one of the things that they are doing at Exeter is offering genetic testing at very low cost or free of cost for those children born in other parts of the world where genetic testing is not available. So thank you.
Thank you very much, Diva. It's an absolute pleasure to be here today. I met some of you in Barcelona last September, and it's great to be meeting more of you today. So I'm a scientist. I did a PhD in genetic toxicology, and I moved to Exeter in the southwest of England in 1995 to set up the genetics lab. Now, that was a really big year for hyperinsulinism, because that was the year that the mutations in the potassium channel genes were found. You understood the cause of hyperinsulinism in many of the children. I have to confess, I was completely unaware of that, because our focus in 1995 was on diabetes. And it was only when we actually found mutations in the same genes causing neonatal diabetes that this area suddenly opens up, and we started doing genetic testing for hyperinsulinism. So in the UK, we have two specialist clinical centres. We have colleagues from Norchai, the Manchester and Liverpool collaboration in the back row over there who you met, and Khalid Hussain and team from Great Ormond Street Hospital. Khalid and I have a research grant from the Medical Research Council, and that allows us to offer the free genetic testing for many patients across the world. And these are some of the people in the laboratory in the photograph who do the genetic testing. And particularly, I'd like to acknowledge the work of Dr. Sarah Flanagan, who's sat just a few rows back. And Sarah now has her own research funding from the Wellcome Trust to take forward the gene discovery work. So the aim of the genetics laboratory is to provide a fast, accurate genetic diagnosis for every patient. Now, it's not possible yet, but that's what we're working towards. And the reason is that the genetic diagnosis will guide the treatment, and secondly, it will define the risk of hyperinsulinism for future siblings and future offspring. And the best example of how the genetic diagnosis guides the treatment is illustrated by these two babies, Jack and George. These are babies from the UK. They were both diagnosed with hyperinsulinism on the first day of life. Neither responded to diazoxide, and they both have an ABCC8 mutation. But Jack's mutation is homozygous, and this told us that he would have diffuse disease, the whole of his pancreas will be affected. And he underwent a subtotal pancreatectomy actually just a few weeks after he was born. Whereas George has a heterozygous mutation inherited from his dad, that told us that it was likely that he had a focal lesion. He had a PET-CT scan that confirmed that was the case, showed where the lesion was, and he was able to have keyhole surgery and came out of theatre cured. In terms of the risk for future siblings and offspring, for Jack, his parents are both carriers of the most common Ashkenazi Jewish mutation. What we mean here is that they have one copy of the gene without a mutation and one copy with the mutation. And they've each of them passed on the mutation to Jack, who's homozygous. And as in any what we call autosomal recessive condition, there's a 1 in 4 chance that the next baby will inherit the mutation from both parents and also be affected. But what we can tell Jack when he's a little bit older, he's only 7, is that there's a very low risk for his future children. He'd actually have to have children with someone who is a carrier of hyperinsulinism for there to be a risk of their children being affected. Whereas for George, focal hyperinsulinism is a very unusual genetic mechanism. I'll talk a little bit more about it later on. So he's inherited the mutation from his dad, so he's heterozygous, one copy of the gene has the mutation. And there's a very low risk that either his future siblings or his children will be affected. There has to be a second independent genetic event. And overall, the risk of him having an affected child or sibling is less than 1 in 1,000. And across the entire world, we've published one family where there were 2 siblings with focal hyperinsulinism, but we're not aware of any other cases, so it's really unlikely to happen, but not impossible. So we ought to add a third child here. This is Gemma, and Gemma has what we call a new mutation or a de novo mutation, sometimes called a spontaneous mutation. And this is what we call a dominantly active mutation. So neither of her parents are carriers of hyperinsulinism. When you look in their blood, you don't find the mutation that we found in Gemma. And what's happened here is that either the mutation had arisen in her dad's sperm or in the mum's egg, so preconception, or else after conception, during the development of Gemma as a developing embryo, there was an event, a mutation arose in her. And what we advise in terms of the risk for future siblings is that there's a very low risk. And that actually depends on whether this was a preconception event in the parent, in the sperm or the egg, or whether it happened in Gemma as a developing baby. And there's new genetic technologies that hopefully will be able to give a more accurate estimate for families. But what's really important to note is that Gemma's future children will each be at 50% risk of inheriting this mutation from her and also being affected. So there are other types of hyperinsulinism, and we'll talk more about this, I'm sure, over the next day or two. In Exeter, as I mentioned, we'd worked on diabetes for many years, and mutations in this transcription factor gene, which is a protein that controls other proteins being switched on or off, were known from 1996 to be a cause of diabetes. And surprisingly, it took us 10 years to realise that actually in some patients they had hypoglycemia in the neonatal period. And this is one of the patients. This is Simon. And he actually was treated with diazoxide for a few weeks. Most of these cases, they're transient. The hyperinsulinism may last a few weeks or a few months. But there have been a few children who have required treatment for years rather than months or weeks. Now here, there's a 50% risk for his future children inheriting the mutation, but it's really important to look out for diabetes because these children would have high risk of developing diabetes either as a teenager or as a young adult. But, really importantly, the diabetes will respond to a specific tablet called sulfonylurea, so they are unlikely to need insulin. Another genetic type of hyperinsulinism are the mutations in the GLUD1 gene that cause hyperammonemia and hyperinsulinism. And most of these patients will again have what we call a de novo, a new mutation, a spontaneous mutation, as I talked about just a minute ago. And this would also be the case for quite a lot of glucokinase mutations that also arise as new mutations. So I'm going to just go through a little bit of sort of introductory genetics, and please stop me if you have any questions. So each of us have 23 different chromosomes, and we have 2 copies of each. So these are our chromosome 11s, and this is where the ABCC8 gene is located. So we get 1 copy of our ABCC8 gene from our mother, and 1 copy from our father. And these 23 chromosomes are packaged up within our cells, and the chromosomes themselves are made up of DNA, deoxynucleic we'll just stick with DNA. It's much easier to pronounce. Here we are, in full. And this is just our genetic code. This is our genetic alphabet. And in the genetic alphabet, we only have 4 letters, which we call bases. We have A's, G's, C's, and T's. And it's the order of these genetic letters that determines the genetic code. And the DNA is arranged into these chromosomes, and you have, as in this cartoon, it describes it as a bit of a long, twisty, rope laddery type thingy. And I've got a question for you, actually. Does anybody know, if you took your DNA and unravelled it, how far would it stretch? Absolutely, yeah, incredibly long, to the moon. The human genome we hear a lot about these days, and it simply means the 3 billion bases of DNA. And you can think about your human genome as being like an instruction manual that allows you to create and maintain a human being from conception until the end of life. And again, this is the question I asked in September, and I have to say, Paul, please don't answer this, because you gave us a really silly answer. So for everybody else, if you typed your genome sequence at 1 base per second, how long would it take? What are we guessing? Years, absolutely. I have to say, Paul just said 3 billion seconds. I was looking for someone to actually calculate. It's actually 90 years. So let's not start off trying to type in our genomes. There's better things to do. So I've got a video here, hopefully. And this is a video that we've made as part of an online course, which is actually about diabetes. But part of the course is just about explaining a little bit about DNA. So I'd just like to share with you this video.
Hello, my name is Melissa Sloman and I'm a clinical scientist. The animation you're about to see is using a cake-based analogy to describe the location of DNA in the cell, how we get from DNA to protein, and what happens when there's a mutation. Each cell contains 23 pairs of chromosomes within its nucleus. Chromosomes consist of tightly compacted DNA. A gene is a segment of DNA containing the code used to synthesize a protein. The human genome contains approximately 20,000 genes. The nucleus of the cell is like a library. The chromosomes in the nucleus are like the recipe books within the library. Each recipe book contains the instructions for making many different cakes, just as a chromosome contains many genes that encode many different proteins. Each recipe or gene contains information about the ingredients and utensils that are needed to make the cake or protein. The exons of the gene are the ingredients needed to make the cake, and the introns are the utensils that are needed to make the cake but don't form part of the actual cake. A mutation is a change in a DNA sequence. The DNA sequence is made up of a mixture of 4 letters or nucleotides, A, C, G and T. The effect of the mutation depends on the type of mutation. The cake recipe states that the cake should be decorated with jam, cream and icing sugar. A point mutation results in a change in the code, so that the instructions now say to decorate with ham instead of jam. This is the type of mutation identified in Jack and Dan, who you met in week 1 and week 2. A deletion of part of the code means that some of the instructions have been removed and now just say to decorate with icing sugar. This is the type of mutation identified in Tom, who has a deletion of 1 amino acid. Some deletions cause the recipe to be completely changed by deleting a letter or letters within a word. The result of the letters are shifted along to fill the gap. The result is an illegible recipe and no cake is made. This type of mutation is called a frameshift mutation. A nonsense mutation means that the protein doesn't get made, and it's equivalent to putting the flour and sugar into the bowl only to find you have no eggs, so it goes into the bin. We hope you've enjoyed the animation, which has shown you the location of DNA in the cell, how we get from DNA to protein.
Okay, I think that's enough. I hope you enjoyed that. This was my backup in case the video didn't work. But essentially, we can think of genes as each being a recipe for a protein or a cake. If we go back to the ABCC8 gene, this is actually a large gene. It has 39 exons, and these are the coding parts of the gene. If you like, the ingredients you need to make the cake. And then you have 38 introns, and these are the non-coding parts. So in our analogy, these are the cooking utensils that you use to make the cake but aren't included in the cake. And the ABCC8 gene encodes the protein, the sulfonylurea receptor protein that Mark showed you earlier. And this is the protein that's critical for controlling insulin secretion. So what usually happens is when you have your lunch, your glucose gets taken up into your pancreatic beta cell and metabolized to produce ATP, and what ATP does is it binds to the channel that Paul was talking about. And this channel is made up of 4 subunits in blue that make the pore, so the middle of the channel through which the ions go through, and 4 of these sulfonylurea receptor 1 subunits. And what this channel does is it closes when this metabolic pathway is active after a meal. It leads to the change in the electrical activity, and then the insulin is released from the cell. Now, when things go wrong, we think about mutations, but we also need to think about this in context. Each of us have 3 billion letters of DNA in our genome, but all of us have a different genome. That is, unless you're a monozygotic twin, an identical twin. And every human genome differs by between 3 and 4 million variants compared to what we use for our genetic analysis, what we call a reference sequence. And many of these variants have absolutely no effect. Other variants define the colour of our eyes, for example, whether you have brown, green or blue eyes. And then there are types of mutations that cause disease. So this child here actually doesn't have any eyes. And this was the video describing the different types of mutations and how they affect the baking of your cake or the production of your protein. And Mark gave us a little bit of information about the consequence of those different types of mutation in terms of what happens to the potassium channel. And when we think about how these ABCC8 mutations cause hyperinsulinism, in some patients these channels just aren't present in the membrane, and that's why the insulin is secreted all of the time and there's not the regulation that there should be. What I'd like to do now is talk about how we actually test for these mutations in the laboratory. And what we do first of all is to extract DNA, shown here from a blood sample, and we use a genetic photocopier to make millions of copies of the genes that we're testing. So for the ABCC8 gene, we then determine the sequence one letter at a time, and we're looking at 10,000 of these letters that make up the ABCC8 gene, and we're looking just for one change in these 10,000 letters. In the laboratory, we use equipment to make sure we have high throughput processes, that we can get rapid results, and that we don't have mistakes in terms of we get the right result for the right patient. So we have a robot to do the DNA extraction. All the DNA samples are in tubes that have barcodes, so we can check we don't have any mix-ups. And all the information is in a password-protected database that's securely maintained. We try and do rapid testing. We test the ABCC8 and the KCNJ11 genes, then encode for the 2 parts of the potassium channel. We try and do this within a week, wherever possible. And if we find a paternal mutation, this would suggest a focal lesion. The patient then would ideally have a PET CT scan to confirm this and to visualize the lesion, and then surgery. If we find a homozygous mutation, this predicts that the disease will be throughout the pancreas, and then the options are medical management or proceed into surgery if that's thought to be the best option for that child and their family. There are some rarer mutations that actually are dominantly actin, so these are mutations where you have one copy of the gene has the mutation, and these are the ones that can get passed on to future generations. And here the options, again, are medical management. And it's the funding from the Medical Research Council that allows us to offer this test for patients who otherwise wouldn't have access to genetic testing. Now, DNA sequencing has come a long way since it was first invented in 1977, where it was a very long process involving radioactivity. This is the fluorescent DNA sequencing that we've been doing in my laboratory actually since we set up. And now we have this new technology, we call it next generation sequencing, that allows us to do DNA testing at a scale we previously didn't imagine. Because instead of testing one gene at a time, we can actually test nearly all of the 20,000 genes in one go. And we do this using a method that essentially uses magnets to pull out the genes we're interested in from the patient's DNA. And this allows us to test for all of the known genes as a second-line test. And we find a mutation in the potassium channel genes in about 30% of the patients who are non-consanguineous, so the parents are not known to be related, and about 49% of the patients who are from consanguineous pedigrees. And you can see the extra pickup rate we get, the extra number of diagnoses we get from doing the extra genes. Now, if we don't find a cause of the hyperinsulinism, we can go on to do exome sequencing. And I'm going to have to skip through some of this because I think I'm a bit short on time. So exome sequencing is looking at the coding regions of all of the genes. And the next step will be to do genome sequencing, where we look at the entire genome. And that's looking to be possible now because the cost has dropped to about $1,000 in the last few years. So what we can do with the new sequencing technology is we can test all of the known genes in one test, and we can use the technology to make new discoveries for new causes of hyperinsulinism. And we can also look in these non-coding parts of the DNA. Now what I'm going to have to do is just to show you the results of this, I think. What we've been able to do is to go on a hunt for looking for mutations in these non-coding parts that are actually 106,000 bases in the ABCC8 gene. And when we went on this hunt, we looked at 2 patients who had focal hyperinsulinism, but we hadn't found a mutation in their ABCC8 or KCNJ11 gene. And deep in the intron, we found the cause. And we were able to confirm this actually affects the splicing. If you remember in the cake analogy, this, for example, was when you were trying to mix up your recipe and you left the spoon in the cake. And we actually found that this ABCC8 mutation is common in patients from Ireland, and we found it now in 6 other patients, and we heard at the scientific conference that the Philadelphia group also have a patient with this mutation. And this was really important for a couple whose first child had actually just died of hyperinsulinism, and we were able to do a prenatal test for them in the next pregnancy and to provide them with the reassurance that their next baby wouldn't be affected with hyperinsulinism. So I just wanted to finish by thanking all of you who've taken part in our research. We've now actually had DNA samples sent from 2,239 patients from 78 countries. Thank you very much.
Thank you. That was fantastic. We have time for a couple of questions.
If your child has de novo, how do you determine whether or not it's dominant or not?
Yeah, that's a really good question. You can't just assume. So what we do is we look at the type of mutation. So the dominant mutations for ABCC8 and KCNJ11 are what we call the missense mutations. And that's where you have a different, you make the protein, but the protein has a different code. And it means that the channel is produced, the channels go to the membrane, but they don't work correctly. And the reason that they're dominantly acting is that each of those channels contains multiple subunits of the protein. And because if you have 50% of the subunits affected in one channel, you might have another channel with 75% of the units affected because the channels assemble randomly. So the net impact is that it's not that half of the channels are affected, more than half of the channels don't work properly. So that's how you get a dominant mutation. Usually, if you find a de novo missense mutation, it is a dominantly active mutation. But we have seen, we had a case of focal hyperinsulinism, and that was actually a de novo mutation. But that was one of these mutations that means that you don't make the protein. So you have to exert some caution, but by and large, it's likely that if it's a de novo mutation and it's a missense type, that would act dominantly.
Any other questions?
Just one question regarding the testing. You stated that you do free testing. We send our blood tests off ages ago when they first were given a grant. Are you still accepting testing, stuff for testing? And if so, can we tell the parents about it?
So the free testing is funded by the Medical Research Council and the Wellcome Trust. We're able to provide that for countries where genetic testing otherwise isn't possible. So for countries like Canada and Australia, we charge for the first test, for the urgent test for the potassium channel mutations. But if no mutation is found, we can offer further testing on a research basis. So it's a slightly more complex financial arrangement, but it's to try and get the best value from the research grant.
Shan, thank you so much. This slide it's just awesome. I mean, the fact that
78 countries. There is testing in 78 countries because of Shan and her partner's work. And for such a rare disease, that number, over 2,000 tests, have been conducted. This work has helped so many children with our rare condition. So we just cannot overstate the importance of this work and of making sure that they can continue to do this work and that we learn from this work. So it's staggering to me. And Shan has already left to talk to the tweens and teenagers and share this information with them.